Alright, so the first thing I'm going to do today, like I said last time, I have 2 open dates, basically for 6 speakers. It's October 25th and November 1st. These are going to be short 10 to 15 minute presentations, basically a practice for your ASA presentation. So I'm going to pass this around, and if you'd like to present on this date, just put your name and your email address, and I'll get in contact with you. Levitt, can you mute yourself?
Yes. It's good.
Thank you. All right. Everybody comfy now? All right, we're good. So today, Dr. Ayre has come to us from Denton, Texas. He is an associate professor at the University of North Texas, where he has been since 2003. He has 3 PhD students currently, 3 PhD students, 2 postdoctoral students, and an opening. If you're a master's student and looking to get your PhD, you can talk to him. He got his PhD in Alberta in plant biotechnology. He did 2 postdoctorates, one at Cambridge in UK doing plant biotechnology, and then another postdoctorate at Cornell where he told me he learned how to deal with real plants. So basically he's done a lot of molecular biology. And so he's going to talk to us.
All right, thank you very much. Thank you very much for inviting me down. Texas A&M, its status and reputation, particularly in plants and agriculture, is one of the best in the U.S. So to be here standing in front of you is it's really quite an honor for me. So let me, here's my title slide. The Phloem Network is a whole plant integrator of developmental signals and nutrient homeostasis. And as I've been talking to people and learning about sort of what the interests of this particular circle is all about, I'm realizing that I have to change my talk a little bit. So I'm going to step away from the more formal presentation that I had originally thought I was going to do. I'm going to basically truncate off the last half of the talk and expand the discussion on some of the earlier slides. So if it seems a little unprepared, it's because I'm changing what I want to present as I meet with new people. So just as a little bit of self-promotion before we move on, this was a recent issue of Trends in Plant Science, and that is a picture of a cotton flower that the postdoc in my laboratory took. There is an associated article with this, but I was not included in the author list, so if you go to look at this subject on phloem mobile signals and flowers and breeding, you'll see the name of my postdoc, but maybe I'm in the acknowledgments somewhere. I don't know. I got left out in any case. Okay, so this was the original overview of what I wanted to talk to you about today. I wanted to give you a quick introduction and a little bit of the history of something that we call phloragen and how it relates to the photoperiodic control of flowering. And then I was going to talk about a technology that we developed at UNT specifically to benefit breeding exercises in the cotton plant, to try to breed day-neutral lines and photoperiodic lines with a little bit more efficiency. Technique that I call virus-induced flowering, or VIF. And then I was going to talk a little bit more about this gene family that flowering locust tea belongs to, and how this gene family contributes to the overall architecture of the plant. So where the flowers are produced, where the branches are produced, when flowering occurs and when flowering stops, and some of the projects that we're doing to move into that area, again with a focus on manipulating cotton plant architecture. Whoops, sorry. These last 2 subjects relate to what the other half of my laboratory does, okay? Basically, I consider myself to be a phloem physiologist, okay? And one half, or almost 2/3 of the lab, focuses on carbon partitioning, the movement of sucrose, the utilization of sucrose between source and sink organs, and it is the other third of the lab that focuses on the cotton flowering. Now that might seem to you a little bit schizophrenic, might seem a little bifurcated, but the central theme there is that we work on processes which are directly related to the phloem. And these last two subjects, okay, I'm realizing as I meet more of you, is that I'm probably just not going to talk about these slides, okay? So we're going to get to a certain point, we're probably going to run out of time, and if you want to talk about carbon partitioning, perhaps we can do that at another time. And since this is a crop and soil, or soil and crop science department, we do see a link between carbohydrate transport and phosphate utilization, but that's more of a physiological nutrient condition more than breeding. So it's interesting, but perhaps not interesting to this group. Okay, so let's take a little bit of a historical perspective on this process of flowering and photoperiodic control. Okay, so origin and photoperiodic control. This dates back to some of the very earliest studies that were ever done in plants, and particularly on the hormone nature of plant development. Okay, as early as 1865, the German scientist Julius Sachs was publishing on essentially a flowering hormone, and just quoting his translated from German, of course, but extremely small quantities of one or different substances arrive in the leaves, and these then influence the plastic materials which we know flow into the vegetative points that they assume the form of flowers. Okay, so this is one of the earliest discussions on what we would now call a hormone. In between the work of Julius Sachs and this guy down here, Mikhail Chalekin, we'll talk about a bit. Very important discovery was made by USDA scientists, where they published the first well-articulated articles on the photoperiodic nature of flowering, okay, working with Maryland mammoth tobacco and also soybean in 1920. And then Mikhail Chalekin, a Russian scientist, is recognized as sort of the father of the origin hypothesis. And continuing on and influenced by the work of Julius Sachs, Chalekin did a large number of experiments where he did grafting between different species, okay, interspecific grafts. And what he was able to show was that there was a highly conserved compound which was able to move from the leaves of induced plants, okay, plants that were induced to flower, and this compound was able to move into the scions of uninduced plants and ultimately cause flowering, okay. He didn't know what this compound was, but he did name this hypothetical morphogen phloragen, okay. He named it basically a flowering morphogen. Now what I want to do is, from 1936, actually before I do that, here's a couple of the experiments that he would actually do. So you've seen experiments similar to this in your textbooks, where essentially what we're doing is we're shading different parts of the plant to ask the question, where is the photoperiodic signal detected, and where is it ultimately, where does it ultimately manifest itself? So these are chrysanthemum plants, and in these different experiments, what he's doing is he's covering different parts of the plant, either the leaves of the plants or the apices of the plants. He's exposing them to different day lengths. Okay, so one part of the plant will be under a long day condition, and the other part of the plant will be under a short day condition, and then watching what the plant does to determine that it is the leaves that detect the signal, and then it's the meristems which receive the signal. Okay. Now this is what the hypothesis that he came up with in 1936. Now this sort of image, of course, it looks very trivial to us now. It looks very primitive. But in 1936, these original hypotheses about the hormonal nature of flowering were wouldn't say I'd go as far as saying they were revolutionary, but they were very important at the time. And so what he's showing here is that if you had a short day plant, under long day conditions it wouldn't flower. Under short day conditions It was the leaves that detected the short day to induce the flower. If you had a day-neutral plant, the plant would flower basically regardless of what the day length was. And in the case of a long day plant, again, the leaves indicated in black are what are detecting the signal, and then it's the apice which is actually doing the flowering. And without that signal, the plant in this particular case would stay in a rosette form and not generate the inflorescent stem. All right, let's jump forward 70 years to when we finally figured out what this Florigen compound was. Okay, so these were two back-to-back papers published in Science, May 2007. And what they discovered was that the protein encoded by heading date 3A in rice and the protein encoded by flowering locus T gene in Arabidopsis, what these compounds were, these were the actual Florigen compound. And instead of being the small hormone-like molecule that everybody thought they would be, Florigen turned out to be a 19 kilodalton protein. And so what it does, and why it took us 70 years to figure out what it is that it's produced in one cell, it moves through the plasmodesmata, ultimately to get to the phloem. It's transmitted through the phloem, and then it continues its symplastic movement to ultimately get to the meristem. So unlike other hormones, there is never an extracellular step involved in this signal transduction cascade. Okay, and that's one of the main reasons why it took us 70 years to figure this out, because regardless of what extracts people made and regardless of how they applied it to the plant, this 19 kilodalton protein was never able to cross the membrane and actually do the signaling that it had to do. Okay. So this is what we understand about photoperiodic flowering today. It's called the external coincidence model, and what I'm representing here is only what's happening in the case of a long day plant, which is a little bit simpler than a short day plant. And in short day plants, it's a little bit more confusing. I'm looking around. I don't see John Mullett here, but he has published some work on short day flowering in sorghum. But essentially what we have is we've got a protein called constans, which is produced in the leaves. Okay, and constans, when it's transcribed following a photoperiodic cycle. Okay, so what happens is about mid-evening, it starts to increase its expression level, and then it drops down again for the day. Okay, now the protein that results from that transcript, it is stabilized by light. Okay? So in a long day condition, when the light is shining, the constans protein is stabilized, and it can then act as a transcription factor on downstream genes. Okay? But under short day conditions, what happens is that the transcript continues to follow its circadian rhythm, but now when the transcript starts to go up in expression, the constans protein, it is translated, but it's immediately degraded because there's nothing there to stabilize it. Okay, so the level of protein is never allowed to accumulate, and therefore it can never act as a transcription factor. Now, downstream of the constans transcription factor is this gene we've already talked about, excuse me, flowering locus T encoding the Florigen protein. Okay, now, when flowering locus T is expressed, the Florigen is produced, it migrates to the phloem, it transports through the phloem, and then when it gets to the vegetative meristem, another signal transduction cascade takes place to ultimately cause the transition of that vegetative meristem into a reproductive meristem. I alluded to these heterografting experiments that Chalkian did back in the 30s. Okay, so for example, he was able to take a tobacco plant that was flowering and graft it to a nightshade plant that was not flowering, and you could get the nightshade plant to flower. So he hypothesized a highly conserved compound. With molecular biology and making transgenic plants, we can go far beyond the heterografting that he could ever do. And this is one experiment showing that the HD3A from rice, if you take the gene from rice, which is a short day monocot, and you overexpress it in a Arabidopsis plant, which is a facultative long day dicot, just the presence of that right FT is sufficient to cause flowering irrespective of the conditions that the Arabidopsis is grown on. Okay, so FT is acting downstream of all this photoperiodic stuff, and if you overexpress it, you can bypass all the photoperiodic control of the plant. All right. This brings me to our interest in cotton. Okay, so as was pointed out during the introduction, I'm a Canadian. We grow precious little cotton in Canada. Okay, I don't use any cotton grown in Canada except for possibly in greenhouses. And when I came to Texas in 2003, I had never even seen a cotton plant before. But when I arrived, I started to ask questions about how I could apply my expertise to a plant that was important to cotton. Okay, so I've learned a lot about this crop over the last 10 years. So cotton is naturally a photoperiodic perennial. Okay, modern lines are day neutral, bred for day neutrality, and we grow them in as an annual. Okay, so this is what we do with modern cotton plants. Okay, we plant a seed in the spring once the soil temperature gets to a certain temperature. Early germination or proper germination is very, very critical for cotton production. So we have to wait until the soil temperature is nice and warm. Shortly after germination, when the plant is about 5 nodes high, the cotton plant will start to flower. And by the end of the season, we've got our crop, and sometime around mid-September or so, we are already taking that crop out of the field. Okay? Now this is what ancestral cotton is doing, shown on the bottom. Okay, it's a perennial. It goes through cycles of reproductive and vegetative growth, and it's a short day plant. So ancestral cotton, the days are short enough around mid-October so that the first flowers that you'll see start to arise around Halloween. Okay, and then because it's a tropical plant, or semi-tropical plant, it's through the mild winters that the flowers mature, the fruit is produced, and then when the days start to get longer again in the spring, the plant goes back to its vegetative cycle. Okay, so it's cycling between vegetative and reproductive growth. Now, modern cotton is highly inbred. Okay, it's gone through several genetic bottlenecks in its domestication. Wild cotton has a vast diversity of genes for resistance to all sorts of things. Okay, but the problem is, if you want to introgress some of this wild ancestral gene, some of this wild diversity into modern cotton, you've got this problem that they're flowering at different times. Okay, and you can use a blackout greenhouse, or you can go down to Mexico and use the winter nursery down there to get these things to flower at the same time. But that's going to increase your costs, and it's going to limit you to annual cycles. And there's also other complications to getting some of these cotton plants to flower. This is just a quick slide that I loaded up this morning, and what we're doing here is I'm trying to emphasize the diversity of cotton that's out there. The cotton that we grow in the United States are specific accessions of these two varieties, Pisidium hirsutum and barbadense. Okay, this is your upland cotton, that's your Pima. And there's a lot of natural diversity out here that could be of great interest to cotton breeders if it was made more available to them. Okay. So the experiments that we started to do at the University of North Texas, we wanted to come up with a way that we could get these plants to flower at the same time, and that's actually shown on the next slide. But before I get to that slide, I wanted to show you the difference between modern and ancestral cotton. So this guy Delta Pine 61. It's not grown currently, but it was pretty popular back in the 60s. It's a relatively small shrub. Normally you would treat this with G8 inhibitors, so out in the field it would be a lot shorter anyways. And over here on this side, we have a accession which is known as Texas 701. The bamboo stake in that pot is 6 feet high. And what we notice here is that this plant, you can't really see it in this picture, but this plant, there are reproductive bolls virtually at each node along the fruiting branch, whereas this one, grown under 16-hour days with supplemental light in the greenhouse, this is completely vegetative. Okay, there's nothing resembling a reproductive bud anywhere on that plant. I'll also at this point out that this variety has this characteristic ochre leaf shape. So it's a very, very palmate lobed leaf shape, and we'll come back to that in another slide. Okay, so this slide essentially summarizes the technology that we developed. Essentially what we've got is we have a virus which naturally infects cotton. It's the cotton leaf crumple virus. It's endemic to the Southwest, okay, so it's already out in the environment anyways. Its symptomology is relatively mild. It will produce a slight crumpling of the leaf, but other than that, it doesn't really affect yield too much. And what we did is we engineered this virus so that instead of having the coat protein gene, it carries the gene for flowering locust tea from Arabidopsis. Okay, so it has the florigen in place of the coat protein. And I'll just mention that the coat protein is required for plant-to-plant transmission. Okay, the virus has to be encapsulated into the coat protein for the virus to stick to the mouth parts of the whitefly. Okay, and if you don't have the coat protein, the whitefly cannot transmit the virus from plant to plant. So the virus carries flowering locust tea. When the virus expresses that flowering locust tea in the leaf, both the virus and the FT protein, florigen, are able to migrate through the phloem, ultimately get to the apices, and cause the signals that cause the conversion of those vegetative meristems to reproductive meristems. This is a structure of the vector that we use. Okay, it's relatively straightforward. I'll point out that we did not actually develop the virus or develop it as a tool. That was done by Rich Tuttle, Candace Hagler, and Nikki Robertson working at North Carolina State University. They developed this technology for VIGS, virus-induced gene silencing. Okay, so their idea was to put genes in the opposite orientation that they would normally be expressed so that they could silence genes in the cotton plants. We're putting the genes in the correct orientation, so we're using it for gene delivery rather than gene silencing. Okay, so does it work? Well, in a nutshell, yes. So what we're looking at on this slide, I'll jump over here to the right-hand side. This is our empty vector control. So this is a plant that's infected with the virus, but the virus is not carrying the flowering locust tea gene. This plant, again, we've got a 6-foot tall bamboo stake. It's completely vegetative. Okay, no reproductive growth. It's 120 days old, grown in 16-hour day lengths. This plant, on the other hand, this was infected when it was very small. We infected the cotyledon shortly after it germinated. And what's represented by these arrows is a small proportion of the very large number of reproductive structures on this plant. Okay, so basically all up and down this plant, we have those meristems trying to develop into fertile flowers. Those that do develop into fertile flowers, they have a very normal morphology. I'll point out the red petal spots that are found in this particular variety. Okay, it's a normal flower. And then what we can do is we can take that fertile flower and we can use the pollen to pollinate emasculated domesticated lines. Okay, so this is our Texas 701 flower pollinating a emasculated Delta Pine 61 flower. The boll grows normally. Inside that boll is the F1 seed. The boll matures, the seed matures, and so from that structure we can harvest our F1. When we look at the F1 seed, we then ask the question, well, do we have effective breeding? Do we really have a cross here? And so this is some of the characteristics which I'll go over quickly of these F1s. This is that characteristic okra leaf shape I was telling you about in the male parent. The Delta Pine 61 has the normal cotton leaf shape, and then the F1 progeny has an intermediate leaf phenotype. Okay, so it's a codominant trait, and it's just phenotypic evidence that we have effective crossing. If we look at flowering time, which again is an important trait in this cross, the Delta Pine 61 flowers at the 5th node. The Texas 701, it's a short day plant. This is long day conditions, so when it gets to node 25, it still has not produced a reproductive structure. Okay, that's what the little asterisk is referring to. It's not flowering by node 25. Then our F1 progeny, again, it's intermediate. It's not flowering as early as the Delta Pine 61, but it is flowering, even under these long day conditions at node 15. This is what the maternal parent flower looks like. There are no petal spots. You remember on the previous slide I showed you the parental male, which did have flower spots. So here is the F1, and it's got the petal spots characteristic of the male parent. Okay, so those are just 3 readily scored phenotypes showing that we've got a successful cross. And what I want to do is emphasize in this PCR is that the F1 progeny is free of virus and is free of any recombinant material. Okay, so the virus that we use to induce the flowering in the first place, it does not enter, there is no recombination between the virus and the genetic material of the plant. Okay, and in a lot of plants and a lot of virus interactions, the virus is not transmitted through the germline. Okay, so if you want virus-free plants, taking it to seed is usually a pretty good way to get that. So what this slide is showing us here is we've got, I think there's about 10 different F1 progenies there, and we are screening for evidence of recombinant virus by PCR. Okay, and we're not seeing any here. If we screen our controls for the rep protein of the virus, it's clearly detectable. Okay, and this bottom strand here, this bottom lane, this is a magnesium chelatase from the plant, just showing you that the PCR has actually worked. So what this means is that, yes, we have used a recombinant virus to induce flowering in the first place, but the F1 progeny that result from this cross are completely free of recombinant DNA. They should not be considered GMOs. Okay? So for, say for example, for an organic breeding program, or for countries where GMOs are not allowed, this is still a viable technology for introducing foreign germplasm. Now, we're not the only people pursuing technology like this. This is a publication that came out a little bit before our publication, actually, where it was a Japanese group. This is some experiments that they were doing in soybean. I'm not going to go through the details of it, but it's essentially showing that they can take photoperiodic biennial soybean and basically accelerate flowering to overcome the environmental stimuli that otherwise would be required. And this, I think, is the more significant work from that same laboratory, that same Japanese group. And what they're doing here is the virus that they're using is apple latent spherical virus. That's the vector. We use cotton leaf crumple virus. They're using this apple vector. Apple and a lot of other fruit trees are particularly problematic for breeding because they have a juvenile period. Okay, so in the case of apples, it's 3 to 7 years before you even get a flower. Okay, so if imagine yourself being a student working on an apple breeding program, it's going to be a long degree, right? Okay, but what they've done here is they've infected the juvenile apple with their virus. They've got a solitary flower there. Maybe that would actually produce a fruit. It wouldn't be a very productive fruit. But the point is that they can take that very early forming flower, they've got functional pollen, and they can use that pollen in some other aspect of their breeding program. Now, where are we going with this at the University of North Texas? There's a lot of plants out there that do not flower when we want them to flower. Okay? And this florigin compound is very highly conserved. Okay? The same gene that works in Arabidopsis will work in rice. So a couple of things that we're currently pursuing. Sugarcane is a very important biofuel crop. Sugarcane breeding is a nightmare because it has a very, very specific environmental conditions. I think there's one place in Hawaii and one place in Brazil where people can get this thing to naturally flower. Otherwise, they have to put these things on a flatbed truck and drive the entire flatbed truck into an engineered growth chamber. So I've heard, I haven't actually seen it, but that's what they do in Australia apparently. And so a virus-induced flowering technology in a plant like sugarcane could be very, very beneficial. Similarly, this is a crop which is not particularly important here in the United States, but it's very important in particularly poor tropical areas. This is cassava. Cassava, it's mostly the root that we're interested in, but there's been very, very little breeding effort in cassava. Okay, and the flowering time and even the nature of flowers is kind of weird in cassava. Okay, so we've teamed up with a group at the Danforth Center in St. Louis, Missouri, where they have a large, many large projects in cassava, and we're going to see if this technology can do something to benefit cassava breeding. These again, I have absolutely no results to tell you about any of these things. It's just an indication of the directions that we're taking it. Now what I want to do is I want to shift gears a little bit and broaden the discussion on what these genes are doing. Okay, so I've talked about one gene, flowering locus T, encoding florigin, and how it can contribute to getting a plant to flower. Flowering locus T belongs to a larger gene family which we refer to as the sets genes. Okay, this sets refers to the genes in different plants that they were first characterized. Okay, so centroradialis in Antirrhinum, terminal flower in Arabidopsis, and self-pruning in tomato. These are all parts of the same gene family. Now, what's kind of interesting about these is that the FT-like genes, these two clades shown down here at the bottom, they are genes that when overexpressed, they promote determinate growth. Okay, so if you overexpress these guys, you take a very, more or less a very vegetative plant, and you can make it more reproductive, or at least smaller in its stature. The terminal flower genes, these are all named after the original mutation, and genetics is always confusing because the name of the gene is named after the phenotype rather than the wild type function. So terminal flower gene, when it's mutated, it produces a terminal flower, but when you overexpress it, it causes vegetative growth. So you get a very, very bushy plant. Just as a couple of examples of this is terminal flower in Arabidopsis. So this is normally what Arabidopsis does. If you knock out or mutate terminal flower, you get, well, as the name implies, you get a terminal flower. So instead of a continuing racemic inflorescence, you get one flower at the very end. This is the same gene, centroradialis, in Antirrhinum. In the wild type, you have a raceme. If you knock out terminal flower, that raceme terminates in a terminal flower. So we have this gene family, very closely related sequence, but the different family members are doing opposite effects. Okay, and some collaborators that we have in this is from a review paper we published, but it's based on a hypothesis that was developed by some collaborators that we have in Israel. And it's a hypothesis for a universal paradigm for how plants control their determinate versus indeterminate growth. Okay, their determinate growth or their reproductive growth versus their indeterminate or vegetative growth. And the hypothesis says that in a very young plant, what happens is that at the apice, you've got a lot of terminal flower expressed. Okay, so you're overexpressing terminal flower, which means you have signals for vegetative growth. Okay, so the meristem is very indeterminate. Meanwhile, flowering locus T is naturally expressed in the leaves. It might be turned on and off by environmental signals, or it might be expressed at all times. But in a very young plant or in an uninduced plant, you have very little FT expression, but lots of terminal flower expression, so you have vegetative growth. Now, as the plant ages, what happens is that the level of terminal flower expression is naturally repressed. Okay, so the signal for vegetative growth or indeterminate growth naturally declines, and in the meantime, we have more and more leaves. The leaves are producing more and more flowering locus T, which is causing an amplified signal to move to that vegetative meristem. And at some point, you get to a tipping point where the determinant signal outweighs the indeterminate signal, and that's when you get conversion from a indeterminate to a determinate meristem, or from a vegetative meristem to a reproductive meristem. What is the evidence for this? Okay, well, when we talk about the evidence for this, we can go all the way back to 1927 and the tomato plant. Okay, the tomato plant, its architecture is surprisingly similar to the cotton plant. Okay, they both produce what's known as a cymose inflorescence. In ancestral tomato, and the type of tomato that you grow in the backyard, it's an indeterminate plant. Okay, it's indeterminate in the sense that throughout the entire growing season, it will produce some flowers and some fruit, but you always have an apical meristem which is vegetative, so you are always getting more and more vegetative growth to produce that growth, which is great in your backyard because you can go out throughout the summer and pick off fresh tomatoes. Now in 1927, an allele was identified called self-pruning, which is the same as terminal flower in Arabidopsis. And what this allele does, what this mutation does, is it causes all of these indeterminate meristems to convert to determinate meristems. Okay, now why do you want to do that? Why is that good? In industrial grown tomato, what you want to do is you want a synchronized crop. You want a flush of flowers, a flush of fruit, all maturing at the same time, and then you go out into the field once and you harvest all at the same time. Okay? So with the development of this self-pruning allele, this revolutionized, or at least significantly changed, the way that tomato is grown in the United States. And today, virtually all, if not all, industrial tomatoes contain this self-pruning allele. Oh, and this is basically a little cartoon of what's happening here. This is your indeterminate tomato that you have in your backyard. So the apical meristem is always producing new vegetative growth. This is the self-pruning mutation where that apical meristem eventually terminates into a final inflorescence structure. Okay, so all your vegetative apices eventually convert to reproductive nature. Can we do the same thing in cotton? Okay, cotton it's a dry fruit. Okay, so getting it out of the field all at once isn't as important as it is in tomato. But notwithstanding, the physiology of cotton is such that the earliest forming fruit has the greatest quality with respect to fiber, and the late forming fruit has weak fiber. Okay, so the late forming fruit is detrimental to the quality of the cotton, and so there's some interest in there about getting the plant more synchronized and getting an earlier phenotype. So these are some of the phenotypes we've seen in our cotton plants when we've overexpressed flowering locust tea. This is what wild or normal cotton does. So the fruiting branches are these cymose inflorescences. When we overexpress FT, we see some interesting things. So this funny looking structure here, it never grew into a proper flower, but it is the plant's effort to produce a reproductive structure. And what it's doing is it's producing it directly on the main stem, where normally cotton would put a branch, a fruiting branch in that location. Okay, so we've overexpressed the flowering locust tea signal to such an extent that the meristem that's in that location has become determinate before it was able to grow out into a branch. Here what we've got, let me just back up a little bit. Normally what happens is that the terminal bud of the sympodial unit, that terminates into a flower, and it's the axillary bud that continues the vegetative growth. Okay, that's how we define a cyme. In this particular example, what's happened, or what we think has happened, is that the apical bud of that branch and the axillary bud of that branch have converted to floral fate at roughly the same time. So what we've done is we've terminated that branch with a floral cluster rather than continued vegetative growth. And essentially the same thing has happened here. It's hard to see it, but there are 3 flowers in that terminal cluster. And what's happening here is that both the terminal bud and cotton actually has 2 axillary buds at each node. And so what's happened is that both of those axillary buds have converted to a floral fate. So we have a total of 3 flowers at the termini of that branch. In domesticated cotton, this overexpression of these determinant genes, they also have the effect of making the plant smaller and more synchronized in its production, okay, which, depending on which physiologist you're talking to, these are attributes for cotton production. This is untreated cotton, and what I want to emphasize here is that with the circles we have a lot of relatively mature bolls, and what they are doing, they are going to produce the highest quality fiber, but at the same time on the periphery of the plant we've got a lot of new growth which is producing new flowers, and what the producer will do is leave the plant out in the field to try to mature these flowers off so that yield is increased at the possible cost of quality. In our FT overexpressing plants, on the other hand, what we've got is we've got vegetative growth, we've got a flush of reproductive growth, and then all of those branches that normally would produce more flowers, they have all terminated into these floral structures. So we have synchronized the crop by preventing formation of new flowers. Now the first thing that you're asking, first thing you're going to say to me is that that's a smaller plant. You're not going to get as much fiber out of it. Okay, that's fine. You plant more per acre. Okay, so when we're talking about yield, we're not so much interested in yield per plant, it's yield per acre, which is the important characteristic. Okay, this comes down to the idea that flowering locust tea is not just a flowering hormone, it's an overall determinacy hormone. Okay, it promotes determinate growth. So what we're looking at on this slide is we're just emphasizing that because we've got this very characteristic ochre leaf shape. When we infect with our virus, we change the leaf from a leaf is always a determinate structure, but this is a relatively indeterminate leaf, and we convert it to a very determinate leaf with a very lanceolate structure. Okay? In the uninfected plants, we do not see this. But when we take this perennial plant and we go from long day conditions to short day conditions and back to long day conditions, we do see this transition in the leaves of going from relatively indeterminate to more determinate to indeterminate. Okay, so we're not This is a natural process which the plant does anyways. This slide, these are our collaborators in Israel who are basically pursuing very similar things in tomato. In fact, what they do in tomato, we are basically trying to see if the same thing happens in cotton, with the idea, are these actual universal paradigms that can be applied to any plant, or are they specific to one or the other? Since this isn't our work and since it's all published, I'm not going to go into too much detail here, but essentially this paper is showing that if you overexpress the FT ortholog, or if you knock out the terminal flower ortholog, you get these same characteristic, more determinate plants. So instead of a flowering truss with multiple fruits on it, you get a single flower, and instead of very complex compound leaves, you get relatively simple flowers, simple leaves by manipulating these ratios. Okay, and with that, the last part, this is the sucrose transport stuff that I've opted not to talk about. There's actually more slides on that than I thought there would be, so I will end with my acknowledgement slide. The collaborators on the cotton work, Nikki Robertson, Kader Haig has been fantastic. It's not very often that the granting agency actually helps you focus your grant, but Cotton Inc. will do that, and Kader has been really great at focusing our thought processes. Eliza Lifshitz and Yuval Ished are our collaborators in Israel, and most of the work that I presented was carried out by Dr. Roshi Magari, a postdoc in the laboratory, and the cotton work was funded, Cotton Incorporated, yay, and USDA. More recently, we have money from the BARD program, which is a collaborative effort between the USDA and Israel. And with that, I'll be happy for questions or discussion. Yes, here.
So, can you suppress PFL1 genetically and get the same effect, or at least something similar?
That's definitely the hypothesis, and the current work we are doing now is exactly that. So we are, what we've done as part of the BARD research is we've taken the 8 sets gene members from cotton. There are 8 of them. And we are using our Nikki Robertson's virus system to both overexpress them and silence them in the cotton plant. And then we are also taking the genomic sequences from cotton and putting them into Arabidopsis. And so our objective is to characterize the gene family, and downregulating terminal flower is definitely a priority, and we do expect it to be equivalent to overexpression of FT. Yes.
This virus transmitted Floridan gene, once you transfer this in Chardonnay plant, does it breed through there after that, or it is just one generation?
In the virus?
No, the plant. The plant has got the gene. Would it breed through thereafter?
I'm sorry, I was a little bit distracted.
You take a Chardonnay plant.
The short day plant, right.
And introduce the Floridan gene through the virus.
Through the virus, right.
And you showed that in the first generation, then you make the cross. But if you grow the progeny out of it, would it breed through? So you have a isogenic line now of Chardonnay and neutral.
No, it would still be short day, because the virus is only transient, and so the gene of the FT, barring any epigenetic effects, let me put that caveat in there, okay? But if it is a purely genetic event, the gene that caused it in the first generation will be gone in the second generation because it's not carried through.
Okay.
So if, for example, you wanted to propagate that line, you could always infect it again with the virus, and then the third generation would again be virus-free and DNA-free. So does that answer your question?
No, you showed the F1 plant
F1.
was intermediate.
Yeah.
So if you self F1 plant, would that now segregate into the true breeding?
Well, the F1 is not flowering because of the virus. What we've done is we've crossed a day-neutral plant with a short-day plant. And so we've introgressed the photoperiodism, or the day neutrality, And when we take it to the F2, yeah, we would anticipate that that would segregate out all over the place. I'm sorry.
So it has been integrated, the gene then has been...
No. Let me just go back to the cross.
Just allows you to cross.
So the virus is only effective in the generation that you do the cross, and I understand this was one parent day neutral, so early flowering. This was basically not flowering under long day conditions. So the reason why the F1 is flowering early is not because of the virus is no longer there, but because whatever is causing this to be photoperiodic or non-photoperiodic, whatever is causing this to be day neutral, a certain amount of that is now penetrant in the new generation. Okay, so this would be the heterozygous condition of these two parents, and then if we took that to the F2, we would expect it to, if it's a single gene trait, we would expect some progeny to be flowering like this, and we would expect some progeny to be flowering like this, and then some to be continuously intermediate. So it's not, we haven't bred anything in per se with the virus.
So I got a quick question just about kind of, I guess the potential costs or labor savings by doing this kind of virus-mediated introgression just versus classical pedigree breeding. We're doing some similar work with a barbiton succession where we're trying to introgress thrips resistance into a kind of a cross between upland and Pima, but the most thrips resistant succession we had is, of course, photoperiodic as all get out, right? So I'm just curious, like, I mean, how much do you save as far as like time and labor, that kind of deal, doing this method versus kind of the old way?
Right. Well, I can't give you actual numbers.
Right.
Yeah. But okay, so thrips resistance, that's really important. You should come to our greenhouse sometime.
Sounds good.
So to do it the traditional way, if you have a really photoperiodic line, you take it to the winter nursery in Mexico, and there you're limited to annual cycles if it is only photoperiodism. If there's a juvenile period, or if you need a certain temperature regiment, it might be difficult to get that guy to flower even at the best of times.
Yeah.
The other alternative, and we've had some discussions with some of the people I met here this morning, is that you do it in basically a blackout greenhouse. Okay.
Right.
That's going to increase the cost of production. You have to make that greenhouse. Even then, you don't know exactly when it's going to flower. The people who read my USDA grants, they must work with a Arabidopsis because they tell me to put it in a growth chamber.
Right. Yeah.
Obviously, never worked with cotton. So neither of those things are required with the virus system. Now, another advantage of the virus system is that if you have to grow your plants under short days, you have less photosynthetic energy, and if you're doing it in the winter, you also have the sun's lower in the sky. If you use the virus, you can grow the thing under 16-hour days, the middle of the July sun, and so the plants do visibly, very visibly, just grow a lot faster, which is an attribute.
Yeah.
Some of the problems that would be associated with it are, first of all, it is a recombinant virus, and it is a virus. So some sort of containment, at least in that first generation, would still be required. And secondly, right now it's not as reliable as we would like it to be. We've talked to Jane Deaver a fair bit about this. She has sent us some photoperiodic lines. We've tried a little bit. We've got very limited greenhouse space at UNT, but we've tried some of those lines, and it's rather sporadic. And how many flowering plants we get, and in some of her germplasm, and this is still her pseudum, some of that germplasm just didn't flower at all. So, in terms of, I've been talking to people, and I'm very interested in getting germplasm that. I'm not a breeder. I don't really know much about cotton at all, but I'm really interested in getting germplasm from people who have interesting photoperiodic sessions, and then just trying it. And then if it flowers, we can send you the plants, or we can send you the virus, or something can happen at that point. Does that answer your question satisfactorily?
Yeah, it does. That's really great. It's really, really fascinating. Appreciate that.
I like his attitude. Yeah, do you have a question?
So in maize breeding, one of the things that's really revolutionized is going from one generation a year to 3 generations a year. So how fast do you think you could push the plant, first of all? And second of all, do you think there's epistatic effects then that might be able to help you push it a little faster?
The epistatic effects, we've seen some things in the greenhouse, but the greenhouses, they're crowded and stuff, and we're a bunch of Arabidopsis molecular biologists, so we're kind of looking for epistatic effects. We haven't seen anything obvious. With respect to the generations per year, we are changing when the plant flowers, but we are not changing anything after that. So from the time that reproductive meristem forms to the time you get an open flower is 20, 30 days. And then to mature that off to seed, you're talking about another 50 days or so. Does that sound right? So you're still talking about 80 days to go from that initial reproductive meristem to a seed. And if you add on to that another couple of weeks before you can actually get, we have never advanced it beyond the 4th or 5th node, so we have not accelerated the earliest of the domesticated lines, and even then it takes about 2 weeks before you get to that 5th node. So doing the math, I think you'd probably still be looking at about an absolute minimum 100 days, which you could get 3 generations in principle in 1 year. You had a question in the back?
Yeah. What's the benefit of using the virus versus transforming with agrobacterium?
You've never worked with cotton. No, that's a very important question. And there are people who work with fruit trees, for example. They have made transgenic fruit trees overexpressing FT, and they use that as one of their breeding lines so that they can basically introgress fast flowering. They call it the fast track technology. So they can introgress a fast track technology into their breeding lines. So it is done in both ways. What's the advantage of the virus? Let's focus on cotton initially. The only line of cotton that will transform is Copra 312. The virus in principle, and Copra 312 is day neutral to begin with, so it's not going to help you. But the virus in principle can infect any accession. Its host range is actually broadly through the malveolés, so you can infect a multitude of different plants. And I think a really big attribute, particularly in these days of Greenpeace and so on, and people not really wanting transgenic plants, cotton, it's not such a big deal, but in other plants, other crop species, you might want to do this breeding without generating a transgenic intermediate, and that's another advantage of the virus. So there's probably others, but I can't think of them right now. But
just dealing with all the security issues and biosecurity and so on. It's a big advantage.
The disadvantage is that the virus is intrinsically variable in what it does. And so, some of the experiments we're trying to change cotton plant architecture on a more permanent basis, and it's really hard to get sort of anything quantitative because there's so much variation. So some of the work that we're moving into, we have to take a transgenic approach because we've kind of reached the limits of what we can do without a transgenic. But that's more for basic science.
Brian, how are you introducing this virus into the plant? Is it at a very early stage?
It is. As soon as the seed germinates and almost before the cotyledons are fully mature, and certainly before the first true leaves come out, we are injecting an agrobacterium solution into the airspace of those cotyledons. If we wait too long, the infection efficiency drops dramatically, and I think that's what's happening, and probably similar to that, why we're not getting infections in some varieties is that the plant just naturally builds up resistance to the virus. Okay, so any cultivar which has a strong resistance to the virus in the first place, that's going to hamper our efforts. So if the plant gets too old, I think it has too much natural resistance. I think she wants to cut me off.
It's 20:05 now. So if y'all have any more questions, I'm sure Dr. Ayre would be happy to answer them for you.